Um, Good afternoon, everyone. Uh, my name is Hyobin Jeong, currently working at the Department of Systems Biology, Yonsei University. And it's my big pleasure and thank you for the organizers to invite me to present our work in SNU GMI International Symposium. Today, I want to talk about the single-cell multi-omics to understand the intra-tumor heterogeneity shaped by structural variant. If we think about the tumor and cancer patient, there are two types of tumor heterogeneity. We can see the inter-tumor heterogeneity between across the patient and also within the patient. We can observe the intra-tumor heterogeneity. Uh, this phenomena, in this phenomena, we can see the distinct cell populations within the tumor that has different epigenomic and genomic properties. This is shaped by multiple cell intrinsic factors such as genetic variation and epigenetic alterations, and this is known to contribute to therapeutic failure and cancer progression. So it is important to understand the intra-tumor heterogeneity to better understand the cancer and also the treatment options. In terms of genetic variation, structural variation, in short, SV, is a key mutational process in cancer together with a single nucleotide variant. SV can be many different categories such as inversion, duplication, translocation, and deletion. And it can lead oncogenic fusion, oncogene amplification, enhancer hijacking, and also tumor suppressor deletion. In our group, we are very interested in this question, how SVs affect epigenomic heterogeneity in aging and cancer. To be able to answer this question, we need a single-cell genomics technology where we can detect the structural variant in individual cells. So for this purpose, today I want to introduce you the technology StrandSeq and also the computational pipeline SC-Trip to analyze StrandSeq dataset. StrandSeq is one of the single-cell DNA sequencing methods that can resolve individual homologs by sequencing only the template strand of the DNA during the DNA replication. To do so, we firstly culture the cell with the BRDU. Then BRDU will be incorporated into one of the strands of our double helix. It will make a hemi-substituted sister chromatid. And after the mitosis, we can see 4 different segregation patterns. Watson, Watson, Watson Crick, 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 and Crick Watson. So this is a very useful when we analyze the data and because after the sequencing, we can align the read to the reference genome. And based on the alignment direction, we can classify the read into Watson or Crick. And in the normal situation, we will see one of those 4 different patterns when there's no structural variant. However, when there is a structural variant, we can see the strand state changes as exemplified in this figure for instance, heterozygous inversion or homozygous inversion. You can see the strand state changes in this point where I indicated with my mouse point. This method is very useful when you have a single cell sequencing because without PCR amplification, the genetic material from single cell is very small. So it's hard to obtain a high depth from the single cell DNA sequencing. But this strand state will give you the information of breakpoint even without the coverage of breakpoint. So we can obtain the structural variant profile even with a very low sequencing depth. So this is an advantage of strand-seq. And I'm showing you the strand-seq result of example single cells. This was profiled from the T-cell acute lymphoblastic leukemia sample, and each page shows the single cell profiles. With eyes, we can already observe some of the strand state changes, and this is the indicator of the presence of structural variant. If we analyze this data using the SC3 program, we can computationally call the breakpoint, and then we can reconstruct digital karyotype. For instance, in this slide, I'm showing you the digital karyotype from the patient 33 of TALL sample. You can see that we can give information of haplotype reserved structural variant, including deletion, duplication, and also translocation. Additionally, strand-seq, because strand-seq is a single cell technology, this is useful to see if there's any subclones within the same sample. For instance, in the patient 33, all the single cells we sequenced mostly shared the similar SV profiles. However, in another patient, patient 1, when we sequenced 79 single cells, we could see different patterns in some of the single cells. For instance, I made the red box here, and this is around 23% of the cell population, which additionally gained very complex structural variant in chromosome 6. So with this, we can reconstruct the subclonal structure within the sample. So far, I showed you how to identify structural variant using strand-seq and SC3 program, and how to reconstruct subclones within the sample. Afterwards, we wanted to know how those different clones are distinct in their functionality. Especially, I wanted to know what kind of pathways are differentially activated in different clones. To be able to know the functional difference, we need to measure the genome and epigenome from the same single cell. So as a powerful solution, we focused on the nucleosome profile as an epigenomic readout. This is the strand-seq protocol I briefly showed you in the previous slide. And you can see that in the step 12 to 18, MNase digestion is involved, which is known to cut the nucleosome-free region of the DNA. As a result, after the sequencing, we will obtain the read from the nucleosome-occupied region of the DNA, as you can see in this MNase track. This pattern is opposite with chromatin accessibility, which we can measure by DNA-seq, pair-seq, or ATAC-seq. So in summary, with the nucleosome profile, we can detect closed chromatin and nucleosome-occupied region. In fact, when we looked at the strand-seq profile from the lymphoblastoid cell line, we could see the peaks which correspond to the nucleosome positions. And this was very well correlated with the position we detected from the conventional MNase sequencing data from lymphoblastoid cell line from the ENCODE database. Using this property as a feature set, we can construct a machine learning model to infer expressed genes and also to infer altered gene activities between subclones. Because high nucleosome occupancy correspond to low expressed genes or unexpressed gene, and low nucleosome occupancy corresponds to the highly expressed genes. So this enabled us to make a model to infer expressed genes and altered gene activities. Based on these properties, we devised the computational pipeline SCNOVA, which is single-cell nucleosome occupancy and variant analysis. We start from the strand-seq data and using this to identify haplotype-aware structural variant and also to measure the haplotype-aware nucleosome occupancy in the gene locus and cis-regulatory element. To ultimately investigate local and cis effect of SVs and also global and trans effect of SVs. The overall goal is to understand functional consequences of genetic structural variant within the cancer samples. And we want to further use this analysis to know the perturbed network in different subclones and also to suggest druggable pathway that can suppress the aggressive clonal expansion in the subclones. Now we are applying this method to different biological questions, and today I'd like to introduce more about the studying mosaic structural variants in complex karyotype acute myeloid leukemia. Complex karyotype AML is defined by the presence of 3 or more chromosomal aberrations. This is the second largest cytogenetic subgroup of AML, which comprise 10 to 12% of entire AML patient. And this is associated with complex chromosomal rearrangement, intra-tumor heterogeneity, therapy resistance, and extremely poor overall survival. So until now, still people are investigating this CKAML to find the better treatment strategy. So in this project, this is showing our cohort. In total, we collected samples from 8 patients, CKAML patients, and then performed strand-seq and also site-seq from the 8 patients. Among those 5 patients, we also we could obtain PDX samples to see the long-term long-term effect, and then we also profiled the strand-seq from these 5 PDX samples. Additionally, among those patients, 2 of them we could obtain matched relapse and refractory samples, and we also profiled strand-seq and site-seq. Firstly, we focused on identifying structural variant from each of the patients, in total 8 patients, and I'm showing you the heat map from chromosome 1 to 3 to X. And this is each patient, and the number within the parentheses. This shows the good quality single cell libraries in each samples. And the color code is here. Sky blue is deletion, heterozygous deletion, and dark blue is homologous deletion, and then purple is duplication, and so on. You can see that because it's complex karyotype AML category, most of the patient shows a lot of rearrangement in their genome. Some of them are shared by all different cells within the sample, but some of them are specific to a single cell or specific to a certain proportion of the cells. For instance, in this CK282 patient, in chromosome 9, you can clearly see distinct population in this area. And also in the chromosome 12, you can see the ongoing genome instability. And if we enlarge the chromosome 12, you can see that very complex chromothripsis event is ongoing in this chromosome. This is a Sarcos plot of CK282 showing the translocations. By this way, we firstly profiled the structural variant in individual patient. Afterward, we wanted to categorize these 8 patients based on their subclonal structures. Some of them shows mostly monoclonal growth in these 2 patients. And then we also see linear growth in 3 different patients. Linear growth means the cells obtain certain structural variant, and some of the cells additionally obtained further structural variant. And then the rest of the patient, 3 patients in this case, showed branched polyclonal growth. Sometimes the structure was very complex in CK282. In total, 5 subclones we could detect. And then we wanted to know the differential gene expression or pathways within the same patient when there's branched polyclonal growth. So we made use of strand-seq and also site-seq to resolve this question. For instance, in CK282 sample, this is the subclonal structure based on the strand-seq data. And as we know this structure, we could assign site-seq data to one of the 5 subclones based on copy number inference. And you can see here, this UMAP is showing the site-seq data, and some of the cells were assigned to SC5, SC1, and SC2, and so on. Interestingly, among those site-seq subclones, subclone 2 and 3 shows high expression level of ATP5mg, ATP5mf, and also the oxidative phosphorylation gene sets, which is the well-known indicator of oxidative phosphorylation in mitochondria in cancer cells. And also in the next page, we applied the similar analysis to the CK349 sample, where we defined 3 subclones based on the strand-seq data. In the UMAP of site-seq, you can see some of the cells were assigned to SC1, SC2, and SC3. Among those cells, SC3, which was quite small in terms of cell fraction, it shows a very high expression of PRDX1, LDHA, and ALDH1A1, which is the indicator of the elevated oxidative and metabolic stress in cancer. This was to understand the subclonal structures and their different pathways within one sample in certain time point. And now we further compared the subclonal composition between the primary sample and PDX sample for those 5 patients where we had matched primary and PDX pairs. Among the 5 patients, 2 of them showed a dominant clone evolution pattern. That means the subclone which was dominant in the primary sample was still remained to be dominant in the PDX sample. On the other hand, the other 3 patients showed minor subclone evolution pattern. That means the subclone that showed very minor cell fraction in the primary sample showed dramatical expansion in the PDX sample. As you can see in the CK2A2, subclone 2, which is dark green, was expanded a lot, and SC1 in CK349, SC3 in CK349, which is dark brown, was expanded dramatically. And also SC3 in the CK397 was expanded dramatically. So within this patient, we could see different pattern of subclonal evolution. Among those, when we observed minor subclone evolution, we wanted to know what's the drivers or distinct pathways in the subclone that eventually expanded in the later time point in the PDX. So in this case, we made use of SC-NOVA, the nucleosome occupancy analysis, to compare the different pathway activities. In the right side, the heat map shows the activated pathways in each of the subclones, and you can see the subclone 2, which was expanded, showed very active MAP kinase signaling, PI3K, AKT, mTOR signaling, and also pluripotency pathways. And finally, we wanted to compare the samples, the subclonal compositions in diagnosis and relapse samples. So one of the patient cohort, we could obtain the time course data from diagnosis to relapse. And then using the strand-seq, we could dissect the subclonal composition. In this case, subclone 1 was largely expanded in the relapse sample after the treatment. And the relapse sample showed a lot more structural rearrangement in the chromosome 6. Also, we could assign those single cells into one of the likely cell type among hematopoietic stem cell and multipotent cell and common myeloid progenitor and GMP and MEP using the nucleosome occupancy profile. And we could see that compared to diagnosis samples, relapse samples show a lot more stem-like cells, which is hematopoietic stem cell. So this suggests that the relapse sample looks more like stem-like characterizations. In this patient, we also obtained a site-seq dataset, and this is the UMAP of diagnosis and relapse samples. And we could see that in the relapse sample, the gene expression level of EIF5A and also MYC targets and interferon gamma response oxidative phosphorylations are more active in the relapse sample compared to diagnosis. And EIF5A is a very well-known marker of translation, and this shows translation is more active in the relapse sample, which is one of the important characteristics of active proliferating cancers. So, in summary, using the single cell strand-seq and SCNOVA and also site-seq analysis, we could dissect the intra-tumor heterogeneity of complex karyotype AML, and then we could compare the primary sample and PDX or the relapse sample, the subclonal compositions. And as a further study, we want to increase the size of cohort to see the commonality of clonal expansion, especially in terms of driver pathways and driver genes. And in the second part, I'd like to change the gear a little bit to introduce the study of mosaic structural variant in hematopoietic stem and progenitor cells along aging. In our body, there are cells undergoing a lot of cell division during the development and also aging. Especially the immune cells are produced a lot every day. So the immune cells are produced from the hematopoietic stem cells, and this process is called hematopoiesis. And sometimes when the hematopoietic stem cell acquire new somatic mutation, it can make its clone, and it can lead the clonal expansion. Then we call this phenomena clonal hematopoiesis. The accumulation of somatic mutations in hematopoietic stem cells during aging lead the clonal expansion and linked to a higher risk of cardiovascular mortality and hematologic malignancies. So it is important to understand this phenomena. However, so far, clonal hematopoiesis was mostly investigated at the point mutation level. So in our study, we wanted to know more about potential contribution of somatic structural variant in this process. So the question was how somatic structural variant contribute to clonal hematopoiesis and how the functional consequences of them. To answer this question, we collected the samples from 19 donors in different ages. There was 3 samples donated from a newborn baby from umbilical cord blood, and 16 adult donors contributed bone marrow samples. From this, we performed FACS sorting to enrich hematopoietic stems and progenitor cells, which has CD34 plus positive phenotype. And then we performed strand-seq analysis. After quality control, we could obtain 1,133 good quality single cell libraries from 19 donors. And then we firstly identified the structural variant from single cells and constructed this karyotype showing the somatic breakpoint in 19 individuals. And also we plotted the stacked bar graph showing the composition of wild type cells and structural variant cells within each of the 19 donors. You can see that in the newborn baby or bone marrow from 34 and 47 aged donor, we didn't see any structural variant, but in the donors above age 60, we started to see some of the structural variant, including loss of Y, inversion, and duplications. And then we wanted to perform single-cell multi-omic profiling by analyzing this strand-seq data using SC-NOVA pipeline, where we make use of nucleosome occupancy. Because our cell population, CD34 plus HS species can be in the different developmental stages. Some of them will be very primitive hematopoietic stem cell, and some of them will be a multipotent cell, and some of them will be a little more committed to the lymphoid lineage or myeloid lineage. So by making use of nucleosome occupancy, we wanted to classify those 1,100 cells into one of the 8 different cell status. Because if we can do this, we can maybe compare the cell type composition of wild type cells and mosaic structural variant subclones. To be able to assign the cell type, we firstly made a cell type classifier. In this case, we used the supervised strategy. We firstly generated single-cell MNase atlas from the FACS-sorted pure population from HSC to MEP. And then use this single-cell MNase atlas, we built the supervised cell type classifier, and this was the 900 signature genes used in the cell type classifier. With this, we could assign the cells into one of the 8 status with very high accuracy. After making this cell type classifier, we projected our strand-seq library to this classifier and then assigned the cell type into one of these 8 status. In this bubble plot, you can see the large bubble, which indicate 19 different donors. And within the donors, you see the smaller circle. When there's this dotted line, this means the subclone which shows the structural variant. And within the dotted line, you can see the smaller bubbles with different colors that shows the different cell type composition. And then we compared the cell type proportion of wild type cells versus mosaic structural variant clones. In the right side, you can see the result of statistical testing. Many of the mosaic structural variant clone were significantly enriched in myeloid lineage progenitors such as CMP and MEP. So we wanted to know why mosaic SV clone show cell type bias and what will be the functional consequence of mosaic SVs in the HS species. So we firstly focused on one of the donor BM712 because it showed very complex subclonal structure, and also it showed the cell type bias into HSC and CMP. Within this donor, around 70% of the cells showed the deletion in the chromosome 17p, and subset of the cells additionally showed the deletion in the chromosome 17q. This is the branched subclonal structure, and by using the nucleosome occupancy analysis, we could identify the genes with altered activities. For instance, in the first comparison, we identified the genes with altered activity in the 17p deletion cells compared to wild type. And in the comparison too, we obtained the genes with altered activity in the subclone with 17pq deletion compared to wild type cells. And then we performed functional enrichment analysis of those gene lists, and we can see that subclones with 17pq deletion showed abnormal pathway activity in the mTOR signaling, and also those genes are enriched with the target genes of SREBF1 transcription factor. Interestingly, SREBF1 was located within the deleted region of 17p, and also the mTOR signaling was the downstream target regulated by NF1, which was hit by the breakpoint of 17q deletion. So by linking those pathway analysis to the structural variant location, we could pinpoint a potential driver gene within the structural variant. But because the size of the cohort was very small, we wanted to verify this association between SREBF1, NF1, and myeloid cell bias using the UK Biobank data. So in the UK Biobank data, we could pair the blood cell composition phenotype together with whole exome sequencing data from 400,000 donors. This provided the opportunity to study somatic mutations in relation to blood count. And from this analysis, SREBF1 loss can contribute to a cell type bias in leukocyte, specifically toward monocyte. This was verified in the UK Biobank database. So in this project, in summary, we wanted to explore somatic structural variant landscape from the healthy donors to see if there's any contribution of somatic structural variant to the clonal hematopoiesis. And as a ongoing project in our laboratory, this is more like a plan, but we want to study the role of somatic variant in liver and MSLD, which is the metabolic disease condition, because cell type specific landscape of somatic structural variant and their functional consequences in MSLD and healthy liver tissue is not fully understood. And actually, MSLD is known as increased risk of MASH cirrhosis and even hepatocyte cancer. So we want to further understand the initiation at this early stage if there's any contribution of somatic structural variant in the liver aging. With this, I want to summarize my presentation today. I introduced strand-seq method and SC-NOVA as one of the single-cell multi-omics strategy to identify structural variant in single cells and predict their functional consequences. And this could be applied to the biological questions such as intra-tumor heterogeneity and somatic mosaicism. And in the future, we would like to develop novel computational method and applications to understand the cause and consequences of somatic variants in our genome. With this, I'd like to thank to my previous supervisors because some of the work I introduced today was began in the EMBL where I did my postdoc, and also I'd like to thank to my current lab members, and thank you for your attention. I will be happy to take any questions.
Thank you, Dr. Jeong, for your great presentation. We have one question from the floor, Professor Kim.
Dr. Jeong, thank you very much for your quite interesting tech lecture. So I have one question. I'm interested in the coronal hematopoiesis. So in your talk, so it was quite surprising that the percentage of the somatic structural variation, the percentage of somatic structural variation is quite high compared to the usual coronal hematopoiesis data using the somatic point mutation. So have you ever checked that common point mutation like DNMT3A or TAT2 or PPM1D like that? So my question is that that kind of point mutation occurs with the CA structural variation or exclusively? So some patient or some cell has somatic mutation, point mutation, or some cell has structural variation. That's my question.
Thank you for your question, Professor Kim. Actually, among 19 donors, some of the donors we had whole genome sequencing, but some of them we couldn't obtain that. So we didn't see the full picture. But for instance, we checked the point mutation in DNMT3, TET2, and JAK. And actually, this didn't correlated with the structural variant very well. So we thought that even though the cohort size was small, at least in our cohort, the point mutation and structural variant was quite independent. But I think in the future, we need to investigate in more larger cohort size to really see the test the correlation between them.
Okay, thank you.
We also have some questions online. Thank you for your interesting presentation. You mentioned that SV cells tend to be enriched for stem-like cell types in relapse samples. I was wondering what underlying mechanisms you think might drive this enrichment.
Thank you for the question. So, in relapse sample, for instance, when we saw the site-seq data, in that particular sample, we could see increase of translation-related gene expressions. So in this relapse sample, we thought that one of the driving force is abnormally active protein translation. However, in another donor, where we could see the clonal expansion comparing primary and PDX sample. In this donor, we could see abnormal pathway activation in the mTOR and AKT related pathway. So I think so far we didn't see one exclusive driver, but it looks like it's more patient-dependent pathway activation.
Thank you. We have another question. Given that somatic mutations accumulate with age, what underlying mechanisms could explain the substantial variation seen among people in their 60s? Could this also be related to cell types?
Among people in their 60s. That's very good question. Actually, we don't know a direct answer yet. But of course, we need to check the point mutation as well, because even if some of the donor doesn't have structural variant, they could also have point mutations. So we need to check that. But one of the interesting observation was that one donor, the age was 92, and this donor didn't show any structural variant, even though the age was highest amongst our donor. So it seems like after a certain age, when the people live very longer and they didn't develop cancer, they show exceptionally stable genome. So, however, but we need to collect more clinical data to be able to answer your question.
Thank you. We have one final question. How many single cell in minimum do you need to see SV calls, or you need to assess whether it's statistically significant?
So in our method, we can call the SV in every individual single cells. So the algorithm is based on the tri-channel processing, so depth, strand, state, and genotype. So within one single cell, we can call SV. But afterwards, we categorize the SV calls into singleton, subclonal, and clonal. So generally, if we observe certain SV with more than 2 cells, we categorize this as subclonal. And when we detect the same SV from all the cells within the sample, we categorize this as clonal.
Okay, thank you. So in the interest of time, I think we have to move on to the next speaker. Thank you again, Dr. Jeong.
Thank you.
Thank you.